we are recording. As usual, to claim the credit for this video, you'll need to send me an email after the video is over. In the email, you're going to have a list of the attendance words that I say throughout the lecture, in addition to a screenshot every time I say one of the attendance words. And at the end, the last attendance word, so to speak, will be me asking you to ask a question about the material presented in this lecture. So again, your last attendance word or attendance phrase or whatever is going to be you asking me a question about this material. So pay attention, and that's it. So the first word for attendance, so you're going to need to be ready to take a screenshot. First word for attendance is yellow, right? So when you write down the word yellow, send it to me. Along with the other words, I should see a screenshot of me pointing at this yellow clock tower. And that's it. It is, let's see, 7:25, so you can fast forward about 35 minutes, and then at that point you should be caught up, and that should be when the lecture starts. Morning.
Morning.
Good weekend?
So far, how about you?
Busy. I had a very busy weekend. Nothing but grading and emails. Right. You a little extra credit for being the first one here. Yeah. Excuse me.
Oh my god, I'm so sorry.
For what?
Jim, I'm so sorry, everybody. So, I did back, and I did cardio, and then my buddy came in after I was about to leave. He's like, yo, you want to hit legs? I'm fucking fine. I'll do it.
Oh,
So,
yeah.
my legs are also sore too.
I've been there a
And,
long time, but. Been there.
yeah.
It's good. I just being able to take care of myself.
We kind of started getting to get not on a proper diet. Right?
So you're the second person here. I'll give you also a little bit of extra credit. morning. Morning. Okay, let's see. Brumfeld is not here yet. Champion's not here yet. Gorby, not here yet. Gross is not here yet. Gunther is here. Randall is here. The other Hill is not here yet. Jackson, not here yet. Keys is here. Perez is here. Sigmund, not here yet. Slaughter, not here yet. Smith is here. Stover is here. Tompkins is here. Vanessa is here. Watson is not here yet. Young is here. Zweibeck is not here yet. Lawrence, I just emailed you. Is here. And then we had, let's see, Brumfeld showed up, and so did C. Hill.
That's me.
Yep. You guys are all accounted for. Anybody I missed so far?
Yeah. Maybe that's here. I'll take it to a board there. Got a chair here.
I'll go ahead and start the announcements early because the first bullet point is the same thing I say every day, so I'm not afraid of starting early and somebody coming in at 8 o'clock and missing it. So again, attendance is mandatory and graded. By mandatory, I mean I have to report it to the university for financial aid reasons. Today is the day I have to do it by noon. By graded, of course, is one letter grade, so a full letter grade of your entire score for the course. So if you're missing classes, it would help if you email me and ask, hey, which classes am I missing? Well, hopefully you already know. And then you can go back and watch the videos, assuming the video recorded. Knock on wood, because we're supposedly recording right now. But anyway, you can make up for it by watching the videos. So anyway, that being said, as usual, I'm going to give a list of words. Write them down. Those of you who are here can turn them in for extra credit anytime before 9 a.m. Anybody watching the video will have to turn those in addition to the screenshots. And of course, the last word for attendance, a phrase for attendance, is going to be you asking me a question about the material covered in this lecture. So the first word for attendance is going to be fish. For no particular reason, just the word fish. All right, it's 8. I'll wait till 8:01 before we officially get started. Some other old news, so I don't mind saying this too before we officially get started. Lab 3 is past due. Lab 4 is past due. Better to turn it in late than never. As we've established after the first week, I remember at first I said if you didn't do the pre-lab, you can't come to lab. But I just changed that because too many people are not doing the pre-lab. So if you didn't do the pre-lab, yes, you can come to lab, but you'll have extra work to do because it's not fair to those of your classmates who did do the labs and who are going to be doing the work because they will be doing the experiments and you will have to watch. But the good news is, speaking of labs, and it is 8:01, so now I can give you some new information. There are no pre-labs this week because the next lab we do is going to be the photosynthesis respiration lab. And that lab is really important because you're going to have to write a lab report. So because it's so important, next week for lab, all we're going to do is meet, and you know how I give you a pre-lab lecture? This time, that whole day is going to be nothing but a pre-lab lecture. I'm going to go really into detail about how that lab's going to work. I'm going to teach you how to do a lab report. So next week's lab is strictly based on attendance. All you have to do is show up on time, and you get full credit. and there's no pre-lab. Of course, it wouldn't hurt if you want to go ahead and start working on the pre-lab that's due next Friday, which is the one for photosynthesis and respiration. So yeah, make sure you get that done. Does anybody have a problem? There's so many people not doing pre-labs. Can anybody volunteer why? Like, are you forgetting? Like, I announce it every time we meet. And anything I can do to help you get those pre-labs done or remind you in different ways? Nobody? Okay. Anyway, speaking of not getting things done or getting things done, exam one, just to remind you, is past due now. Exam one was due Sunday. I mentioned it every time we met last week. I said, hey, exam one's due on Sunday. So 7 of you didn't take it. 1 of you, I know why. But anyway, for all of you, including the people who didn't take it, except one person, they know their story, you can now retake the exam. You can take it as many times as you want. It's unlimited. It's open book. And then the only catch is you have to have it done before exam 2 is due. So in 4 weeks, you have 4 weeks to retake exam 1. And the other thing is you have to remember to email me and say, hey, I'm done, go adjust my grade. Because nothing will happen automatically. And I won't know to change your grade unless you email and say, hey, I'm satisfied with my new grade. And at that point, I will average your first score with your highest score. So you can retake it and retake it and retake it. Like, let's say you get a 98 one time, and then the next time you get a 96. Well, your 98 is going to be the one that counts. So it does not hurt you to retake the exam. Plus, the questions from exam 1 will be used to write the final exam. So exam 1 is also your study guide for the final exam. So yes, you guys need to take the exam. It'll do nothing but help you. Even those of you, again, who didn't take it, you have a 0 right now. But assuming you get 100% on the retake, that will give you a 50. 50 is better than a 0. So also, there was a little glitch in the system that I'm sure some of you noticed. So for those of you who did take the exam on time, you may have gotten a little special surprise that I didn't attend, and I'll just leave it at that. But don't expect that for the other exams. That was a mistake, and I'll leave it at that. But again, if you missed the exam that I reminded you every day last week, you missed something, you missed a good opportunity too. But anyway, any questions about anything? You guys have your own announcements or reminders? All right, homecoming's coming up. As usual, if you go to any kind of WVSU, official WVSU event, including the mini for homecoming, if you don't have to be there and you're there, you take pictures of yourself there, I'll give you an extra minute. I think that's all I have for announcements. All right, let's move forward. So to remind you, we are doing chapter 3, which I'm going through very quickly because, like I said earlier, moving forward, all the way between now and towards the end of the semester, it's all going to be about cells, right? So this chapter is all about how cells function and what their parts are. But that's all we're going to talk about for the majority of the semester. So because of that, I'm going through this quickly because all the important stuff from this chapter you're going to learn again later. So that being said, I'll remind you, last thing we were talking about was plant cells. This is, I believe this is the last thing we talked about. I was talking about this thing called the central vacuole that I'm not going to test you on the exam. That was the last thing we mentioned. So let's move past plant cells and go into animal cells. Animal cells have something called an extracellular matrix. You can see a picture of it right there. It's mostly made up of glycoproteins and collagen that are secreted from the cells. And basically it holds cells together, and that's what makes up tissues, and that's what allows them to communicate. Now, I'm going to put a big old X through that, just as a visual indication to you that I'm not going to ask you any questions about that on the exam. But are there any questions about the extracellular matrix? All right, same story with this next slide. Intercellular junctions. I'm not going to ask you anything about this on the exam. I'm just teaching out of the textbook, so I'm going to mention it very quickly. They are the direct contact between cells for communication, right? So these intercellular junctions are part of the cell extracellular matrix. This is stuff called plasmodesmata. These are the junctions between plant cells. Now we're talking about plant and animal cells. Animal cells contain, or cells contact include tight and gap junctions, things you don't need to know. And these things called desmonies, but I'm not going to ask you about any of this. Here's some pictures of it. I'm trying to get through this quickly so we can get into the important stuff. Now we're starting to get into some of the important stuff. Next, we're going to talk about the cell membrane. So this lab that we're going to do this week is all about diffusion and osmosis, right? And that is used outside of biology, but we're going to be focusing obviously on the biology aspect of diffusion and osmosis. And as far as we're concerned, the biggest thing about diffusion and osmosis is how things come and go between cells. So we have these little fake cells that you're going to use, and the material that they're made up of, as far as we're concerned, are basically the same as the cell membrane. So that's what we're about to talk about now. By the end of this section, you should be able to understand the fluid mosaic model of membranes and describe the function of phospholipids, proteins, and carbohydrates in membranes. Yeah, I'll tell you when we get to the important stuff as far as the lab and as far as the exam is concerned. Let's jump into it. Fluid mosaic model. You might get a question asking about what the mosaic model is. That it describes the structure of the plasma membrane as a mosaic of fluid components. In other words, your cell membrane is not solid, it's not rigid, right? It's fluid. And you have all these parts in it. You can see these pictures, like the glycoprotein that we're going to talk about soon. A peripheral membrane protein, some generic one. All these different things you can see, they kind of move around, right? For the most part, they're not stationary. Sometimes they are, and we might talk about that. But yeah, generally speaking, they all move around, hence the name fluid mosaic model. Animal cells have cholesterol in the membranes. What does cholesterol do? I'm probably not going to ask you this, but just so you know, it helps regulate that fluidity, so keeping it from being too stiff or rigid. But it also works a little bit as antifreeze, which is interesting if you want to write a paper, a research paper about this. When you think about that, probably different species of, well, any organism, not just animals. Well, yeah, we'll talk about animals. We'll narrow it down to that. Any species of animals probably has different levels of cholesterol in the membrane, depending on where they live, right? They probably evolved to have different amounts of antifreeze because, you know, if you live in, I don't know, Alaska, for example, you probably need more antifreeze than some species that lives in the Amazon rainforest. But anyway, I'm probably not going to ask that either. Any questions about that? All right, as you already know, so you do need to know this because I've said it a few times and I'm going to keep saying it a few more times. The membrane is mostly made up of a phospholipid bilayer, right? So that was brought up in the last chapter when we were talking about lipids. And I've already mentioned it a few times in this chapter, and it'll come back and we'll talk about it again later. So yes, you do need to know that. We'll circle there. Membrane is mostly made up of a phospholipid bilayer. And I might ask you this two things, but I did mention it previously in the last chapter when we were talking about the phospholipid itself, right? We were looking at it strictly as a biological molecule. We weren't looking at it as a whole. So strictly looking at it as a biological molecule, the surface is hydrophobic and the inside is hydrophilic. That's why they're attracted to each other. That's why you have that bilayer. It's gonna be a really horrible drawing. If you had two phospholipids, right, you got that hydrophobic on the outside and those two fatty acids, and those are attracted to each other. But anyway, definitely need to know that the membrane is mostly phospholipid bilayer, and you might need to know that the surface is hydrophilic and the inner part is hydrophobic. So any questions? All right. So, like I said, the membrane is mostly made up of phospholipids. But what's the second most prominent portion? And that's proteins. I'll put a question mark next to that. I might ask you that too. So what is the fluid mosaic model, right? If we're talking about cell membranes, again, you might get a question saying, what is it mostly made up of? It's mostly phospholipids. What's next, right? Second most, proteins. So you might need to know that. And that kind of makes sense too. Remember back when we were talking about biological molecules, we were talking about proteins, and I said when you take the water out of a cell, proteins make up a majority of the cell. So it's kind of makes sense that proteins would be the second major component. They do a few things. I'm almost definitely not going to ask you this necessarily, but we will talk about these individually. For example, sometimes these proteins act as channels or pumps. Because remember, again, the cell membrane needs to control what comes in and out of the cell. Some things can just come right in and out through the membrane. Some things need to be controlled, and that's what these channels and pumps are for. And there's also these enzymes. You're going to learn more about those later in the semester. Enzymes basically just make things happen, specifically chemical reactions, but everything that happens in cells is a chemical reaction, basically. So enzymes make everything happen. There's also the attachment for the cytoskeleton, which, as far as we're concerned in this class, is very unimportant because we'll, for the most part, not going to bring that up again. And it also helps with cell recognition, which, again, for this course is unimportant. None of those four things are unimportant as far as life is concerned, as far as our cells are concerned. As far as this semester is concerned and understanding concepts later in the semester and things you're going to need to know for the exam, these two bullet points are definitely more important than those two bullet points. So again, we're gonna learn all about channels and pumps and enzymes. Especially in this chapter, we're really gonna learn about these channels and pumps. Because again, it's all about what's coming in and out of the cell. So any questions about this slide? All right, the next word for attendance is going to have an arrow pointed to it. Maybe 2 arrows pointing to it. There's 2 arrows pointing to the next word for attendance. So obviously, if you're watching the video, when you send me a screenshot, in addition to the list of words, I'm going to see a picture with arrows pointing at it. So no questions so far? All right, so this is the second most major component of the cell membrane. Next, we're going to talk about the third. Third, does anybody guess? Think about the, I almost said it, so if you have the sharp ear, you might have heard it. Think about the 4 biological molecules that we learned about last chapter, right? So we've gone through 2 of them. We've talked about lipids and proteins. So this is a multiple choice question with two choices. Actually, yeah, it makes it easier when I put it that way. What do you think the third major component is? Again, think about the biological molecules. There's only 4 categories of them. We've only talked about 2. So the other 2, really, one of these options is definitely not going to be in the membrane. So what do you guys think? This is a good time for me to slow down anyway. Think about the 4 biological molecules. We've talked about lipids. We've talked about proteins. What are the other 2 that are left? You guys being shy? Good, carbohydrates, right? Because the only other 2 left are carbohydrates and nucleic acids. And obviously, we're not going to have DNA or RNA embedded in our cell membrane. Good job. If you send in your extra credit words today, remind me that you answered that question. I'll give you a little bit more extra credit. So yeah, carbohydrate is the third major component. These happen to always be found on the exterior. They're always bound to lipids or proteins, so they're not just sitting there floating around on their own. And they are used for cell recognition. I'm going to put big old X's through this, question mark next to this. If I ask anything, it'll be that you need to know the third major component. And I'm probably not going to ask you any of this other stuff. Again, for life, we need all this stuff. All these 3 other bullet points are important. But for this class, it's not important because we're not really going to be dealing with this stuff in this semester. So that's why I'm saying it's unimportant. It's unimportant for this course, not for life. So any questions? We're done now talking about the cell membranes. Are there any questions about the cell membrane? All right, now we're going to get into some really important stuff as far as the exam is concerned and as far as the lab is concerned. We are going to talk about passive transport. If I remember correctly, that's a word that was not in the lab, and I should probably add that to the lab now that I think about it. Because really, this is what we're talking about. So, as you should know from the pre-lab, membranes are selectively permeable. And I've already said that before, I just never use those words. And when I say I've said that before, what I mean is I've talked about how the cell membrane needs to control what comes in and out of the cell. So that's part of how it does it. It's selectively permeable, meaning some things can come in and out, some things can't. Here's a picture of something, for example, and this will make more sense later in this chapter. Here we have a protein that just kind of lets things through. It's like a hallway. As if this was a wall, these things couldn't get through the wall. That's just a hallway for them to go through. On the other hand, we have this, and again, you'll learn more about it later in this chapter. These things, again, can't go through the membrane, so they need a way to get through. In this case, there's this thing. It's almost like a pump. So yes, it's not just a hallway where it can just go through. This actually requires energy. But again, we'll talk about that later. The point here is, again, membranes are selectively permeable. You need to know that. You need to know what that means. Which is why you had all those seemingly repetitive hypothesis questions in the pre-lab, because I wanted you to think, all right, is starch going to be able to come out of the cell? Is iodine going to be able to come into the cell? Is salt going to be able to come out of the cell? Some of those answers were interesting, but we'll talk about it in lab. If you download the PowerPoint, you can watch this little animation about the selectively permeable membrane, about how it'll let some things in and not other things, and let some things out and not other things. Also, here's another little video you can watch if you download the PowerPoint. So here we go. Now the important stuff as far as the lab and the exam is concerned. Molecules vibrate and wander randomly. I don't remember if your textbook uses this word or not, but hopefully you're familiar with it from the lab, but that's Brownian motion, right? So here's a good picture, not that you need to study this, but you can see this is a water molecule. Just one single water molecule, and you can see all the different ways it can move. Like in this case, I wish you could animate it. So imagine this thing with the arms kind of going in and out, and that going up and down, right? And in this case, this one's coming in while that one's going out, and vice versa, right? This one, it's kind of moving like it's flapping its wings. This one's rotating this way. This one's rotating that way. This one's bending this way. So it's all different kinds of ways a molecule can move. And that's just one molecule, right? This is water. So imagine something like that in 3 dimensions. I know it's tiny and we can't see it in real life, but those are what the molecules look like. And they hit each other, right? That's Brownian motion. So when they hit each other, that causes them to move around, and that's what causes diffusion. Diffusion is the movement of molecules from an area of high concentration to low concentration until equilibrium is reached. Remember some version of that definition for the exam. You need to know some version of that definition. When I say some version, it's a multiple choice exam, and I'm not going to be tricky and have like, oh, here's this version, here's the version that you learned from your lab, and here's a version from some other source. It's not going to be like that. There's only going to be one correct answer. The rest of them are going to be completely incorrect. It's not going to be a trick question. So you need to know what's movement of molecules. That part's important. You need to know the movement from high concentration to low concentration. Or another way of putting that is down the concentration gradient, and I'll put that in writing later. And also you need to know that it happens until equilibrium is reached. Another thing you need to know about diffusion is it does not require an input of energy. Hence, passive transport. This is why, This is why it's called passive transport. Does not require energy. As you might imagine, there is something called active transport that we're going to talk about that does require energy. But yeah, passive transport does not require energy. I'm sure you're all very familiar with diffusion, right? So if I were to light a scented candle right now, these gentlemen up front will probably be the first to smell it way before you guys did in the back. But then it would spread and spread and spread. And at first, it would be really strong, like a strong scent up here. And again, eventually it would diffuse out, and eventually, wherever you went in the room, it would be the equal, what's the word I'm looking for? Equal strength, right? So it'll smell just the same in the back corner as it did up here. That's diffusion, right? Because it's moving from an area of high concentration to low concentration. And once equilibrium is reached, that means wherever you go in the room, it would smell the same, like the same strength of scent. So that's diffusion, or a common example of diffusion. Obviously, when we're talking about biology, we're more concerned about things coming in and out of the cell. All right, and then on the next slide, I'm going to have a little visualization that shows you what's on the words on this slide. Are there any questions about the words on this slide? Okay. So again, a visualization. I think this is from the old textbook. But imagine this is animated and not just a still picture. These things are bouncing around. So imagine just a bunch of balls bouncing around. Then you have this selectively permeable thing. So sometimes some of them are going to hit it just the right spot where they can bounce to the other side. And you can see eventually equilibrium is reached where you have an equal amount on both sides. What's worth noting, not for the exam, just so you know, keep in the back of your mind, just because equilibrium is reached doesn't mean things aren't moving. It just means, generally speaking, it's the same concentration on both sides, right? So you might still have things coming this way and that way, but generally speaking, you have the same concentration on both sides. And of course, if we're talking about a cell, that's what we mean by both sides, right? Maybe this is the outside of the cell and this is the inside of the cell, or vice versa. But the point is, that's what equilibrium is, right? When the concentration is equal on both sides. This next picture adds a second dye to it. The same thing's happening, but of course, in this situation, you had a higher concentration of purple on the left, or blue, whatever color that is, a higher concentration of red on the right, and then diffusion is allowed to happen, and eventually you have equilibrium for both of them, where you have an equal concentration of both on both sides. So any questions about this slide? All right. This is going to be a little bit different than what you learned in lab, and I apologize for that. But here are the factors that influence the rate of diffusion. The concentration gradient. Right, so again, using a scented candle as an example. If I had a tiny little vanilla scented candle, like one of those little teacup things, like that big, and lit it, versus if I had some kind of huge candle, the same scent, with a big wick, right? That's concentration. So the one that's bigger, the one that's more concentrated. That would make it diffuse quicker. The max, and this is what you're going to do in lab, right? So we're not dealing with concentration gradient in lab, but we will deal with mass. Because even though you can't see it, on that first experiment you do, you're going to have a petri dish, and you can put a dye on each side of the petri dish, and it's going to diffuse out. You're going to measure it every 5 or 10 minutes, whatever it is, every 15 minutes. You're going to measure how big that blob is, because the blob is going to get bigger as it diffuses out. And even though you can't see it with your naked eye, those two dyes have different mass, right? So one of them on a molecular level is smaller than the other. And as most of you correctly assumed, the smaller one should technically move quicker, right? Because it's smaller. It's easier for it to get through the agar that's in the petri dish. The next one is temperature. Hopefully, as you've learned from previous discussions, like I mentioned earlier, at the molecular level, temperature is a measure of how fast molecules are moving. So at the molecular level, when we say the temperature is increasing, what we're saying is molecules are moving faster. Temperature is decreasing, we're saying molecules are moving slower. Therefore, obviously, hopefully that's obvious then, as temperature increases, generally speaking, diffusion should happen quicker because molecules are moving quicker. And then, of course, this one's where it gets a little bit different, and I apologize. I'll make sure this is not confusing for the exam. The answer will either come from the lab or from here, and I'll never mix the two, so you have to choose which one is the most correct, right? But anyway, the other one is solvent density. So basically, again, remember, think about you have that petri dish, it's got the gel in it, and you put the dye in there, and it's going to go through the dye. But we're not doing solvent density in the lab, otherwise we would have petri dishes with like really thick stuff in it, and then we'd have a petri dish with stuff that's not so thick, right? So we're not dealing with that. We're dealing with mass and temperature in the lab. Any questions about this slide? All right, if you download this PowerPoint, you can watch this animation about diffusion. Again, we're still talking about diffusion, but to make things a little bit more complicated, you do need this for the exam. But let me hold on, let me take a step back. We're talking about passive transport, right? Passive transport, which means things moving in and out of a cell without the input of energy. That's what we're talking about. Under the category of passive transport is what we just finished talking about, which was diffusion. Now, there's a second category that's also under passive transport, and it's called facilitated diffusion. This is because sometimes things just can't come in and out of a cell. Right here is what we talked about. This is diffusion. This is an area of high concentration. This is an area of low concentration. Whatever these things are, they happen to be able to pass directly through the membrane. So they will. They will move here until equilibrium is reached. These things, for whatever reason, cannot pass through the membrane. Right? There's something about maybe their size or their chemistry, but they can't go through the membrane. So they have these things, which are proteins. And that's what is used for facilitated diffusion, right? It's a protein that just it's like a hallway that lets it through. Like imagine if I filled this room with, 200 people, right? That would be really hard, right? We'd be crammed, and we would want to diffuse out of the room, right? But we can't because the door is shut. Now, that doesn't mean nothing can diffuse in and out of this room, right? Air is coming in and out of this room through the crack in the door. The humans are too big, right? So even though nature would want to let us diffuse out to the hallway, it can't happen unless there's something big enough to let us through. And that would be opening the door, right? Once you open the door, then some of those 200 people will start diffusing out to the hallway. Same thing here, right? So these red things, these little red triangles, that is an area of high concentration. They want naturally to come to this side of the membrane. For whatever reason, they can't make it through, so that's what that protein is for. And that is facilitated diffusion. So again, you definitely need to know that passive transport includes two different types. There's diffusion, which we talked about first, then now facilitated diffusion. And of course, you almost could think of facilitated diffusion also as a subcategory of diffusion. It doesn't matter which way you think of it. There's not going to be any kind of question about hierarchy. The point is, all these things we're talking about, these two different things we're talking about, still fall under passive transport. Excuse me. Let me see, I've got those stars to remind myself. Let me see what they are. Bear with me, please. Also, I need to make sure we're recording. So far, we haven't had a full recording. I think a couple of meetings. All right, we're still recording. Very good. Anyway, Okay, so there we go. Sorry about this, guys. Just want to make sure I'm not missing anything important. Okay, yeah. So what's written here is suffices for studying for the exam. But just so you know, when I say substances that don't cross spontaneously, what I mean again is like using these as an example. For whatever reason, they can't make it through the membrane. But just so you know, I'm not going to get this detailed on the exam. Sometimes they can make it through the membrane. It's really way too slow. So again, you need that extra protein, this little hallway, to make it quicker. So again, sometimes it isn't that they can't make it through, it's just they can't make it through quickly. For the exam, all you need to know is that they just can't make it through. Another thing to point out, just so you know, not for the exam, but when I say they make it through these proteins are also very specific. So maybe I shouldn't call them a hallway, because a hallway, generally anything can come in and out of a hallway. But these proteins are very specific, and they only let specific molecules. So there's obviously a bunch of different types of proteins, because they're going to let through a bunch of different types of molecules. Excuse me, I'm coming down something. Anyway, any questions about facilitated diffusion? All right, let's talk about osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. So in a sense, we're still kind of talking about passive transport. In a sense, we're still talking about diffusion. Now we're just getting really specific. Now we're talking about the diffusion of water. Excuse me. Here's some terms I've introduced you to before, and I said they would come back, so here they are again. So you need to know these terms. A solute is a substance that is dissolved in a solvent, and the resulting mixture is called a solution. So if I wanted to make some simple syrup for my margaritas, I would put in some water, I would heat it up, I would put in some sugar and dissolve that sugar in the water in that case, and then I would have simple syrup. In that case, the solute is the sugar, the water is the solvent, the simple syrup is the solution. Well, I included this little meme that I found that I see a lot because it's incorrect. Because when they're talking about information going from an area of high concentration to low concentration, from the books to the brain, it's funny. I know it's meant to be funny, and I shouldn't take it so seriously, but that's not osmosis, right? Because it's information, not water. So that would be diffusion. That should say, why doesn't diffusion work like this? It's ironic, because whoever came up with that, and I've seen 1,000 different versions of it, didn't get the information. Therefore, they screwed it up. Anyway, any questions about osmosis? We're not done talking about osmosis, so let me rephrase that. Any questions about osmosis so far, or any questions about these terms that you need to know? Solute, solvent, solution. All right. Download the PowerPoint and watch this little animation about osmosis. Let's see who can figure this out. All right. So let's pretend like we're not talking about osmosis. Forget all those words on the screen here. Just look at this picture. If we were talking about diffusion, which way would the balls go? The green balls, would they go from right to left or left to right? We're just talking about diffusion, and that was it. And everything else is considered equal. Which way would those balls go?
Right to left.
Right to left. Thank you. That is correct. Because they want to go from their high concentration to low concentration. So that means we have a high concentration of whatever those things are, and a lower concentration on this side. Here's some more terms you need to know. Not necessarily because I'm going to test you on, ask you what they mean, but there's going to be test questions where I say this thing is hypertonic, this thing is hypotonic. Where's the water going to go? It's basically what's going to happen, right? So you're going to need to know those terms. So yes, here we have a higher concentration of those green balls. Here we have a lower concentration. The higher concentration is called hypertonic. Lower concentration is called hypotonic. Like a kid. When someone says a kid is hyper, what does that mean usually?
Energetic.
Yeah, a lot, they have extra energy, right? They have a lot of energy. So in a sense, when you think of hyper, you should think of a lot. Like when something goes hypersonic, it's going faster than the speed of sound. So hyper basically means a lot. And you're gonna need to know the difference between hyper and hypo, so make sure you understand that. Anyway, like someone correctly said, those balls would want to go from right to left. They would want to diffuse from right to left. However, this thing here is semi-permeable, and it lets water go back and forth, but those green balls are too big. So that being said, and remember this whole thing about passive transport, whether it's diffusion or osmosis, it's all about having equilibrium, right? Equal concentration. So here, that's too concentrated. Here it's not concentrated enough. Which way is the water going to go? Because again, we want to have equal concentration. I'll even give it a number, even though I'm just making it up. Let's say this is, I don't know, 75% green balls, whatever that is. 75% sugar, I don't know. And this is 25% sugar. Which way is the water going to want to go? All right, let's put it a different way. Again, let's pretend like this is a simple syrup, right? It's water and sugar. I don't know if that's what it is, let's pretend. And this is 75% sugar, it's really sweet. This is 25% sugar. Now let's say I gave you a glass of water and said, all right, can you make these a little bit more equal? Like you don't have any more sugar, you can't do anything with the sugar, but I gave you some water. Would you pour water into here on the left or here on the right to make things more equal? On the right, right? Because it's really concentrated. So let me dilute that and put some water in there. That's also what nature's going to do. So because of this, nature, with osmosis, water is going to move to the right. And I'm really taking a lot of time on this because there's going to be a few questions on the exam where you're going to need to be able to problem solve and understand where the water went and equilibrium and all that. So this is why I'm going pretty slow on this. Not only will there be a few questions about it on the exam, but historically, those are the questions people usually miss. So again, I'm slowing down to make sure you really understand this concept. Also, by the way, once equilibrium is reached and both sides are equal, that's what we call isotonic. So again, isotonic means both are equal. Hypertonic means one's higher concentration. Hypotonic means one's a lower concentration. You're going to have a lot of, or at least a few questions where I'll be like, all right, got 3 beakers, and in these beakers is water that 25% sugar solution. And then you're going to put semi-permeable balloons in there. The first balloon is 10% sugar. The other balloon is 25% sugar. The other balloon is 85% sugar, whatever it is. And you're going to need to be able to predict what happens after, a given amount of time. Thinking about this, right? So you're like, all right, this one's hypertonic, this one's hypotonic, so water's going to move into the balloon. This one's opposite, so the water's going to move out of the balloon, right? That's how it's going to go. So you really need to understand how this works. Any questions so far? Yes. Basically the goal, ignore this right side for now. Wait, you want to explain this again, or what I was talking about the beakers? Because the beakers I'll talk about later, because there'll be some actual examples. So to re-explain this, osmosis is all about making things equal. Actually, so is diffusion, right? It's all about having equal concentrations. Here, right, we have a really high concentration of whatever that is. I just called it sugar. Let's just call it sugar. Really high concentration of sugar on that side, a really low concentration of sugar on this side. So this is not very sweet. This is very sweet. I was asking earlier, if I told you to make them equal, and I said there's no sugar, right? If I gave you sugar, you just put more sugar in here, right? If you're trying to make this as sweet as that, you would just put more sugar on this side. Does that make sense? Okay. Now, I'm saying you can't have any sugar. We're out of sugar, but you can have water. So since you can't make this sweeter, you can just pour some water in there and make that less sweet. Does that make sense? Okay. And that's just what nature does. That's osmosis. So instead of you pouring the water in there, water itself is just going to go right through that permeable membrane until it's equal. And that's what this picture is. So water kept moving in there until the concentration of sugar, that's what I'm calling it, those green balls, until it was equal. That's osmosis. Osmosis is when water moves. And of course, when we talk about it, it's not just the movement of water. We're also talking about how it moves until it reaches equilibrium. It's trying to make things equal. And if you have questions, I'll ask again later because I'm going to give some more examples. And then I'll ask if you have questions. But for now, there's that. Here's some words that I talked about. Let's put them in writing. Everything I've just mentioned is called tonicity, which describes the amount of solute in a solution. Probably, I'm almost definitely not going to ask you that word. But again, you do need to know these words: hypertonic, hypotonic, and isotonic. So there'll be questions on this exam where I describe something. I'm like, this thing is hypertonic to this thing. Where's the water going to go? And the water is always going to go towards the hypertonic, right? It's going to go towards that higher concentration of whatever the solute is because it wants to dilute it. Water is always going to leave the hypotonic because once you take the water out of that lower concentration, whatever that thing is, whatever the solute is, that's going to increase the concentration, right? It's all about trying to make these hypertonic and hypotonic things isotonic, which is equal. So any questions about those terms? All right, here's some more terms. This one's a little bit less important as far as the exam is concerned. You will get questions on this, but historically most people get this correct. But anyway, let's talk about it anyway. So for a cell to survive a hypotonic or hypertonic environment, it has to balance how much water is coming in and out, right? So you probably, I don't know, you may have not thought about that before, but your cells are in liquid, like in blood and things like that, and they have to regulate their water balance because if they're in a hypotonic, let's start with hypertonic since we talked about it. If it's in a hypertonic environment, the water is going to want to leave the cell to make that hypertonic less concentrated. Meanwhile, the opposite would happen if your cell was in a hypotonic. If your cell was in a hypotonic environment, that means your cell itself is hypertonic. Therefore, water would want to come into the cell to make it less concentrated. And that's where we get osmoregulation, because otherwise your cells would have really big problems because water would just be coming in and out without any kind of control. So for an animal cell, and I might ask you this, isotonic is preferred, which makes sense because like what did I just explain, right? If a cell is in a hypertonic environment, the water is going to leave, it's going to get dehydrated. If it's in a hypotonic cell environment, the water is going to go into it. But, you know, your cells need water, but there isn't too much of a good thing. So that's why isotonic, at least for animal cells, isotonic is preferred. I should write that, at least A for animal. Isotonic is preferred for animals. Because again, you don't want too much water going out, you don't want too much water coming in. Isotonic, it's nice and balanced. Like I've already mentioned, but here it is in writing, if your cell, I'll try to draw it, your cell is in a hypertonic solution. All right, so let's just say, again, I don't know, let's just talk about glucose. Let's say your cell is 5% glucose, I'm just making up a random number. It's in this solution that's 25% glucose. Right? Osmosis and diffusion, they're going to want to do their thing. So water is going to want to leave the cell to dilute the outside. And that's because that would cause it to dehydrate. Right? That's that situation. The opposite would happen in a hypotonic environment. So again, this time, let's say your cell is 25%. And again, I'm just making numbers up. 25% glucose. Oh, that looks so horrible. That's your cell. And it's in an environment that is 5% glucose. Again, water is going to want to balance things out. So in this case, the water is going to want to go into the cell. And it's going to just swell up until it blows up. And that's what lice is. It's going to explode. So any questions on this slide before I show you the next slide, which will show you a picture of what I'm showing you? All right, actually this next slide shows you a picture. These, I was talking about animal cells. These next pictures are going to be plant cells. Plant cells are a little bit different because they have a cell wall. So in this case, isotonic isn't necessarily bad for plant cells, but for them, hypotonic is preferred because they want cells, water coming into the cell because that'll excel the cell wall. So remember, for animals, that's not good because we don't have a cell wall. Our cells will just explode. But for plants, that's a good thing. And of course, whether you're talking about a plant cell or an animal cell, hypertonic is a bad situation, right? The water is going to leave the cells, it's going to dehydrate the cell, it'll kill the cell. Any questions about that slide? All right, the next slide is not important for questions or the exam. It's just more like relating
what I just taught you to what you already know. As a plant cell loses water, it shrivels. It pulls the plasma membrane far away from the cell and usually kills the cell. You might not know that, but you're familiar with this, right? When a plant gets dehydrated, it gets all wilted like that because there's no more water expanding on those cell walls. So think about it. It's all water. The thing that keeps this all turgid like that, it's just water. You remove the water and it gets all limp like that. Of course, you put water back in and it'll, assuming you do it soon enough before it dies, it'll go right back to that. And that's your next word for attendance is plant. Plant right there. Plant. I know we have one person online for the first time, I think. So just a reminder, if you're online, you don't have to send the screenshots. You can just send the words as long as you send them before 9 a.m. Of course, if you're watching the video at a later date, then you have to send the screenshots. But for you who are watching currently live online, all I need is the list of words before 9 a.m. Anyway, here's everything we said in one picture. Again, in a normal environment, aka isotonic, this is what animal cells prefer. You got as much water coming in as going out, right? Because it's at equilibrium. If we're in a hypertonic situation, hypotonic situation, right? So the inside of the cell is more concentrated than the outside, then water is going to want to go in to diffuse or to make it less concentrated. So much water is going to go in, eventually the thing's going to explode. Of course, if we're in a hypertonic situation, the water is going to leave the cell. It's going to become dehydrated. But in short, for the exam, what you need to know, all that to say, animal cells prefer isotonic, plant cells prefer hypotonic. So there you go. Any questions about that? Ooh, we might be able to knock this chapter out today, which would be a perfect time. We'll see. So again, we're talking about the cell membrane, right? We're talking about things coming in and out of the cell because cells need to be able to control what comes in and out of them. We just finished talking about the different types of passive transport, right? Diffusion, facilitated diffusion, osmosis. None of this requires an input of energy. It just happens. Nature takes its course. This next one we're going to talk about is the opposite. By the time we're done talking about this, you should understand how electrochemical gradients affect ions. I'm going to discuss it. Then we'll be done with it. We're not going to talk about electrochemical gradients for the most part, or ions. It'll come back up in a later chapter, but it'll be a lot more simple than the way it's going to be presented here. You can also be able to describe endocytosis, including phagocytosis, pinocytosis, and receptor mediated endocytosis. So it's a lot of big words, but by the time we're done with it, you should be okay. So again, we just finished talking about passive transport. Active transport is the opposite. It does require energy. You need to know that. Active transport requires an input of energy. Usually, that energy is something called ATP, which you're going to learn about next chapter. Usually, it's ATP. That would be a good extra credit thing. So I'm saying it usually requires ATP. Where else does it get energy? So you could just send me an email, separate email, not one with words, where you just simply answer it. Like, hey, I looked it up. According to this source, this is the other source of energy. That'll give you 1 point. Or if you want more than that, you can write a paper about it, whatever you want. But that's, I think, a good question. Active transport usually requires ATP. As far as the exam is concerned, it requires ATP. As far as you're concerned, you can look up the other stuff if you want. It also requires a transport protein. You definitely need to know that. That's important. This stuff, again, this stuff will be important later in the semester. So this isn't trivial. I mean, none of it's trivial, but as far as the exam and the semester is concerned, some of this stuff is less important. This will be coming back later, so it's an important concept. Active transport moves a solute against its concentration gradient. Remember, unlike passive transport, it goes down the concentration gradient. Here it goes against the concentration gradient, right? You're going from an area of low concentration to high concentration. As you might imagine, that's going to take some energy to do that. And the reason it happens is because it maintains a concentration of small solutes that differ from the environmental concentrations. In other words, your cell's going to need, sometimes your cells just need stuff. And because of diffusion and passive transport and stuff like that, sometimes those ions are going to want to leave the cell, because nature's going to want to take its course with diffusion. Sometimes we need it, so therefore we had to put a little energy to get it, and that's what active transport is. Any questions about that? All right. Pay close attention to that screen one more time. I'm gonna have another screen. I'm gonna have another attendance word on the next slide. When I pull up the next slide and ask you the attendance word, or tell you the attendance word, it's gonna be a question. So I'm about to ask you a question about this slide. There's your heads up. About to ask you a question about this slide. Okay. Next word for attendance. Active transport requires energy. Usually that energy comes from what? Of course, if you're watching the video, you can just rewind it. But for you guys, it's just extra credits, so it doesn't hurt you if you don't get it. Anyway, that's active transport. Really quickly, let's talk about electrochemical gradient. I'm not necessarily going to ask you questions about this, at least not in the context of chapter 3. But this idea of an electrochemical gradient will be important later when we start talking about photosynthesis and respiration. But for now, let's just brush up on the subject. Well, I'll introduce you to it. It's not that important for now. So the interior cell is negative relative to the exterior. I'm definitely not going to ask you that, but just so you know, if you're looking at the cell membrane, the inside of the cell, generally speaking, it's negative compared to the outside. So, long story short, there are reasons other than the concentration gradient that necessitates active transport. In other words, again, when we were talking about active transport, we're saying we're trying to get these things going up the concentration gradient, right? We're trying to get them to go from where they're less concentrated to where they're more concentrated. And the only reason I'm bringing this up is to point out that sometimes it's not necessarily about the concentration gradient. Sometimes it's about the charges. But again, for the exam, this does not matter. The only reason I even brought it up is because this is a 100 level class, and hopefully you guys are reading your free online textbook. I don't want you to read that term and be like, he never even mentioned that. And this is confusing. So now I'm telling you, if you read it and it's confusing, come to me if you want during office hours and I can explain it to you. But don't worry about it because it's not going to be on the exam. If you download the PowerPoint, you can see this little animation about active transport. Yeah, I'll leave it at that. Here's a picture of what we're talking about. You can see we're trying to go from a low concentration to a high concentration. That's going to require some energy. It goes into that transport protein. The transport protein kind of changes shape and spits it out, so to speak. And when we get back, we'll talk about the endocytosis and exocytosis. So that's it for the day. Don't forget the last attendance word is to ask me a question about the material presented. And I'll be in office hours from 8 until 9. You guys have, 9 until 10. You guys have a good week.